recording. Share the screen. I got that sort of hiding there. Okay. So today we're supposed to begin. We're supposed to be on chapter 3. Scroll that up a little. And we're having a quiz this Wednesday. And the quiz will cover chapters 1, chapter 2, the start of chapter 2, in lab modules 0, 1, and 2. Any question about any of that? Lab module 3 will not be on the quiz, and only the start of chapter 2 will be on the quiz. I should ask, is there anyone who had trouble, or still is having trouble, with Respondus Lockdown Browser? I got an email from someone and I don't know if it was resolved. All right, I'm going to assume everything's good. Let's begin. I'm not hearing anything, so hopefully everything is good. Chapter 3. So we had finished talking about bright field illumination. Was that correct?
Yeah.
Thank you. So we now need to turn to electron microscopy, and that uses an electron microscope. I thought we talked about this, did we not? Does this slide look familiar? Nobody's commenting.
No, it doesn't look familiar. I think we looked at it last time.
That slide you have up now looks familiar.
I thought we talked about that. Let me see if I have another 2 here. Let's go back a little. I'm not sure why this isn't working.
I think last lecture you already finished. You were on chapter 4 now. Like you started like 12 or 13 slides into chapter 4.
Chapter 4. Like I said, I thought we had talked about that. This looks familiar. Yeah, let's do the search. Oh, yeah, we talked about motile cells. And that is that cells have 2 types of taxis, chemotaxis and phototaxis. Thank you for whoever was making the statement there that we're in chapter 4. I was on the wrong chapter. Does that mean we're ahead of schedule? Let me look at the schedule here. Oh, I'm thinking that today is Tuesday. Today is Thursday. So that's why we are supposed to be on chapter 4. All right. I was confused. Come on, close. It's not going to close for me. All right. So let's talk about another structure that some cells have, and that is an axial filament, which I don't even use that term. I call them endoflagella. It is a flagella, but it is a special flagella in that it is not external to the cell. In fact, the endoflagella is inside the cell wall. It is found only in spirochetes, and the flagella is anchored at one end of the cell and inside the cell wall. It causes the cell the rotation of this flagella causes the cell to move in a corkscrew-like fashion. Let's see if I can get that to open. We won't watch it right now, but we will shortly. And as you can see, the endoflagella there, it wraps around the cell. And let me see if I can blow this up. When we look at it this way, you can see that it's inside the cell wall. It is a little bit of a extension of the cell wall, meaning it makes the cell wall a little bigger, but it is wrapped by the cell wall, shown in the pink. And I don't know, the red is the axial filament. Any question about any of that? Well, let's see where I've got my video. Here it is. And you can see, let's stop this and start it again, that it moves in a corkscrew-like fashion, and it's flexible, like it bent that way and then started moving in a different direction. And you can see how it just changes direction by changing the way it's rotating. So this one is moving down. I think this is it. Unfortunately, it doesn't show it change and start moving that way, but it can just by switching its rotation. Any question about any of that? Oops, wrong one. So the spirochetes have endoflagella, and there are special flagella inside the cell wall. It allows the cell to have a special movement, and it's thought that some of the parasitic spirochetes can actually use the corkscrew-like movement to burrow through the mucous membranes and then infect our tissues. Any question about any of that? All right, another structure that some cells have is a fimbriae. It is shorter and straighter and thinner than the flagella. It's used for attachment to surfaces. It is not used for swimming. Okay, so the cell cannot use the fimbriae for moving when we're talking about the cell swimming through a liquid media. You don't need to know this, but the cell can use the fimbriae for moving over a solid surface. And that is, let's do it from this side. What it can do is put out the fimbriae here, attached to the surface, and then pull in the cell, pull the cell this way and pull up the fimbriae on this side. And then it can put out the fimbriae again in front of it, and then pull the cell forward. So it can use the fimbriae to pull itself on like a solid agar culture. But we're not going to talk about movement in that sense. When we're talking about bacteria movement, we're talking about the ability of the bacteria to swim through a liquid culture. And I was just letting you know that the fimbriae can be used for movement on a solid surface. But we're not going to talk about movement in that way. The pili, or the sex pili, is longer than the fimbriae, and it is hollow. And it is used to transfer DNA from one cell, this cell, which makes the sex pili, into this cell. And the sex pili literally skewers this cell. And then the cytoplasm from this cell can move into that cell. Usually it's just a plasmid that moves across. Any question? we'll talk a little bit more about the sex pili near the, well, later in the term, I think. It looks like I've got a little bit in the end of this class assignment, but it's mostly later in the term when we're talking about genetics. The prokaryotic cell wall is a structure that is important for all bacteria, at least if the bacteria has a cell wall. The cell wall plays the same role in a bacteria as it does for a plant cell, meaning the cell wall is complex, semi-rigid structure, and is responsible for the cell shape. The cell wall protects the cell and prevents the cell from undergoing osmotic lysis. Without a cell wall, water can move into the cell, and if there was no cell wall, the cytoplasm would then expand as more water moved into the cell. Eventually, so much water would likely move into the cell, depends on what environment the cell's in, but in a hypotonic environment, water will move in until the cytoplasm will literally burst. And if you've ever put red blood cells in a beaker of water, you'll notice that within a minute, the blood in the water, the color will disappear from the red blood cells lysing. And what that is simply water moving into the red blood cell and then lysing the blood. Now, for prokaryotic cells, those that have a cell wall, when water moves into the cell, the cytoplasm will expand only up until it's touching the cell wall. After it expands to that point, it can't expand any further because of the protection of the cell wall. And that will prevent more water from moving in because the cytoplasm cannot expand any further. Any question about any of that? The cell wall is located outside the cell membrane, and even though the cell membrane is considered the gatekeeper of what can come into and outside the cell, we do consider the cell wall the last part of the cell. Anything external to the cell wall we call external to the cell. The cell wall can contribute to the pathogenicity of some species, like Mycobacterium. The portions of the cell wall can contribute to the pathogenicity of that bacteria. The cell wall is mostly made up of peptidoglycan, at least in bacteria. The main molecule of the cell wall of bacteria is peptidoglycan. Any questions about any of that? Peptidoglycan is a repeating polymer of two saccharides, so we call it a repeating disaccharide, and the names are NAG and NAM. Let me get this over here. NAG is N-acetylglucosamine, and NAM is N-acetylmuramic acid. And you can use either one because I'm sure both mean about the same to you. But this is NAM, followed by NAG, followed by NAM, followed by NAG, and it just repeats. And then peptidoglycan is a polymer of these two disaccharides. And we have one polymer chain lined up this way, and then a chain on this side and this side. And then underneath this, what do you call that, plane, there are other planes of peptidoglycan. Chain specifically of the repeating disaccharide NAG and NAM. The NAG and the NAM are linked with short peptides, and there will be a cross link, which is best shown here, and this is holding that chain to that chain. But if you notice, this peptide connects to this peptide, which is holding this chain and that chain, and then it's hooked to this chain, which is holding that chain and that, it's hooked to this peptide, which is holding that chain to that chain. Anyways, that's the way peptidoglycan links, holding the glycans together. And the sugars are called glycans, and there are peptides in the glycans, and that gives the name peptidoglycan. Any questions about any of that? Penicillin actually inhibits the lattice formation of the peptides. So penicillin, how it acts is, when the cell is putting down peptidoglycan, before it's linked this chain to that chain with this peptide, the penicillin can get in this region and then prevent a peptide from forming here, linking this chain to that chain. That will create a gap in the cell wall where this chain isn't linked together with that chain, and so we'll have a hole in the cell wall, and the cytoplasm can protrude out of that hole, and as more water flows into the cell, the cytoplasm will protrude, or the cell membrane will protrude further and further until the cell membrane will eventually burst, in which case the cytoplasm will then leak out of the cell and the cell will die. Any questions about any of that? All right, let's look at the cell wall in a little more difference, and that is a little more detail. There are two types of cell walls. There's a gram-positive cell wall, which is a thick purple layer on top of the yellow layer being the cell membrane. And as you know, that's a lipid bilayer, and we'll talk a little bit more about that later in the lesson. When we look at that peptidoglycan layer, all it is layer upon layer of the two sugars hooked together with the short peptide chains. And that's the main molecule of the cell wall. In fact, in gram-positive cells, there's only a few other molecules, and we're seeing one here, lipoteichoic acid and teichoic acid. Teichoic acid runs through the peptidoglycan, and it's thought to help stabilize and hold the peptidoglycan together. A lipoteichoic acid does the same, meaning it runs through the peptidoglycan, but the lipoteichoic acid has a lipid region which also binds to the cell membrane. All right, any question about any of that? It is thought that ions can use the teichoic acid, and I suppose the lipoteichoic acid, to help the ion move through the cell wall. So the teichoic acid does play a role, but it's a minor role because an ion will be soluble in water, and anything that's water can go right through the cell wall, at least of a gram-positive cell wall. Any question about any of that? Nobody's going to ask, well, how does it move into the cell then? You can't get through the cell membrane. So how does an ion that the cell wants, like maybe sodium ion, how does it get through the cell membrane? Come on.
Through like channels that are in the membrane?
Yeah, a protein channel or a transport protein in the membrane will allow that ion to move through the the cell membrane. And each molecule that the cell wants to either enter the cell or leave the cell through the cell membrane, the cell has to have a protein to move the molecule across, either as a transport protein or a channel protein. And generally those proteins are molecule specific, meaning they only allow one molecule to either leave the cell or enter the cell. Any question about any of that? All right, if not, let's talk about the gram-negative cell. Oh, there's a little bit more about the gram-positive cell. Sorry. It's just showing you the teichoic acid, which I've already discussed. So let's move on to the gram-negative cell. The gram-negative cell wall is different. It does have the cell membrane, the yellow layer right here, which is a lipid bilayer, and then it has a cell wall on top of it. And you'll notice it has a purple region, which is kind of thin, which is the peptidoglycan layer. And then on top of the peptidoglycan, it has another yellow layer, which is another lipid bilayer. Now this picture is showing you only one layer of peptidoglycan in the cell wall of a gram-negative cell. That's an oversimplification. The peptidoglycan layers are much thinner in the gram-negative cell than in the gram-positive cell. Where do I have that? Right there. So it's much thinner, the peptidoglycan layer, but it is not just one layer of peptidoglycan. It's multiple layers of peptidoglycan. In the gram-positive cell, we may have more than 100 layers of peptidoglycan. In the gram-negative cell, it's not that many, and it's much less, but it's more than one. And let's say it's a few to, I don't know, a dozen or 2 dozen layers of peptidoglycan in the gram-negative cell. So this picture is an oversimplification. It's not one layer of peptidoglycan, but it's many less than in a gram-positive cell. Okay? Also in the cell wall of the gram-negative cell, we have a lipid bilayer on the outside of the cell wall. Let me blow that up a little. Well, actually, my next slide's bigger. Let's talk about the lipid bilayer of the gram-negative cell. This we call the outer membrane of the cell wall. It's only found in gram-negative cells. This layer of the bilayer is familiar to you. And if you notice, this layer of the outer membrane looks the same as that layer of the cell membrane. And in fact, they are very similar. The main molecule of the cell membrane is, of course, phospholipid, and that is what is making up the bottom bilayer of the outer membrane. The top bilayer does have phospholipids in it, and those are the yellow molecules with the 2 legs going down. But it also has a blue molecule, which is lipid A. So that's a lipid. And then it has a O polysaccharide attached to the lipid A, and that's the hairs you're seeing here. So together, the O polysaccharide and the lipid A make a lipopolysaccharide. Any questions about that? The lipid A is extremely important because lipid A is an endotoxin. And that means anyone who has a gram-negative infection is going to be exposed to an endotoxin. And we'll talk a little bit more about toxins later in the lab. But toxins are toxic substances, and they're generally bad. And the patient, if he has a gram-negative infection, is going to have to deal with exposure to the endotoxin. Any question about any of that? Now, the outer membrane of the cell wall does have other proteins in it, a little bit different than the cell membrane. And that is, the outer membrane has the porin protein that makes a channel. And anything that the cell wants to either enter the cell or to leave the cell has to leave the outer membrane of a gram-negative cell through the porin protein channel. So for example, any amino acid that the cell is going to want, or any sugar, it has to move through the porin protein channel to get through the outer membrane, because this lipid bilayer will prevent most molecules from crossing. Anything that's water soluble will not get through this lipid bilayer. It is true that oxygen and carbon dioxide can get through it, and we'll talk a little bit more about those later. The porin protein is different in that it allows many different molecules to come through the outer membrane. So the outer membrane is not, as particular about what it allows in and out. In the cell membrane, to get through the cell membrane, you have to have a specific protein to move that molecule either into the cell or out of the cell. For the outer membrane, almost everything is handled by the porin protein. Any question about any of that? All right. Let's move on. So the gram-negative outer membrane has a lipopolysaccharide. It does have lipoproteins, and of course phospholipid being the main molecule in the outer membrane. You don't need to know this. Where is the picture of that? But there's a periplasm. Is that the earlier one? There it is right there. And the periplasm is where the peptidoglycan is found in the cell wall. The gram-negative outer membrane can protect the cell from certain things, such as complement proteins and antibiotics. If the antibiotic is binding to the peptidoglycan, for example, or to the cell membrane, the outer membrane will act as a barrier to keep the antibody from binding to the cell. If you don't know, antibodies are a protein that our bodies make for defense, and the important part for a gram-negative cell is if they have the outer membrane, that outer membrane can help protect them from complement proteins, which are involved in our defense, and from antibiotic. The outer membrane can also protect the gram-negative cell from penicillin G. Penicillin G cannot get through the porin protein or through the outer membrane. And if you remember, I mentioned that penicillin acts on the peptidoglycan. And so penicillin G, if it can't get to the peptidoglycan, it's not going to work on a gram-negative cell. Any questions about any of that? The O polysaccharide on the attached to the lipid A can give the cell antigenic variation, meaning our immune system can recognize that O polysaccharide. And when we talk about E. coli O157H7, O157 is the specific O polysaccharide antigen on this E. coli cell, meaning that it's 0157 is this part of the cell. I already mentioned lipid A is an endotoxin, and so that makes a gram-negative cell, when you get infected by a gram-negative cell, an important event because you're exposed to an endotoxin. All right, let's take a little different view and take a look at the differences between a gram-positive cell wall and a gram-negative cell wall. The gram-positive cell wall has a thick peptidoglycan structure, and it's a rigid structure. The gram-negative cell wall has a thinner peptidoglycan layer, and it's not as rigid. It still is rigid, and the cell wall can protect the cell, but it's just not as rigid as the gram-positive cell wall. You can find teichoic acid in gram-positive cell walls. There's no teichoic acid in the gram-negative cell wall. There's no outer membrane in the gram-positive cell wall. There is an outer membrane in the gram-negative cell wall, and that's important because in the outer membrane you can find lipid A, which is an endotoxin. Any question about any of that? All right, let's talk a little bit about the gram stain. The gram stain happens because of the gram-positive cell wall and the gram-negative cell wall. Crystal violet, which will complex with the iodine, forms in the cell. And in the gram-positive cell, the crystal violet iodine complex does not leave the thick peptidoglycan layer. In the gram-negative cell, the crystal violet iodine complex washes out of the thinner peptidoglycan. So that thin peptidoglycan layer makes these cells stain gram-negative in the gram stain. And the alcohol, decolorizing step will remove the peptidoglycan, and it will dissolve the outer membrane of the cell wall. And then this cell will be clear, and then we stain the cell with safranin, and the gram-negative cell will then stain pink from the safranin. On the other hand, the gram-positive cells continue to stain purple because they contain and retain the crystal violet iodine complex. Any question about any of that? All right, so this table gives you a nice summary of the differences between the gram-positive cell, thick peptidoglycan in the gram-positive, thin in the gram-negative, teichoic acid present in gram-positives, absent in gram-negative. You don't need to know about the periplasmic space. Outer membrane absent in gram-positives, present in gram-negatives. The lipopolysaccharide, which is a part of the outer membrane, is none in the gram-positives, and it's fairly high content in the gram-negatives. You can ignore the lower portion of this table. All right, so we've talked about cell walls in bacteria. Let me talk about some prokaryotic cells which have atypical cell walls. And I should probably have this one first, because I should talk about it first. The archaea are not bacteria. They belong in a different domain, and they have different cell walls. They lack peptidoglycan. They do have cell walls, and the cell wall is either made of pseudomurine or the archaea lacks the cell walls. You don't need to know about pseudomurine, it's another disaccharide. The point is the archaea have different cell walls than the bacteria. Any questions about that? There are two types of bacteria which have atypical cell walls. The mycoplasmas are different bacteria, and they have atypical cell walls because they lack a cell wall. All they have is a cell membrane. And like our cells, which are animal cells, they have sterols in their plasma membrane to give the plasma membrane strength. If you don't know, our plasma membrane has cholesterol in it to give the plasma membrane strength. Any question about any of that? All right. We will hardly talk about the mycoplasmas in this class, but they are atypical. They are a bacteria, but they lack a cell wall. The Mycobacterium is another family of bacteria which have atypical cell walls. They are gram-positive, but they are different. And that is, they have a lot of mycolic acid in their cell wall. Mycolic acid is a lipid, and for this reason we say the Mycobacterium are acid-fast, meaning they'll stain with an acid-fast stain. They will stain gram-positive with a gram stain because they are gram-positives, but they are a different type of gram-positive, and we call them an acid-fast cell. Let me see if I can show you a picture of a myco. Oh, that's the lab. I don't want that. Give me 1 minute here, please. I'm not seeing a picture of that. All right, I'll try and find that later and show you later, because I'm not finding what the mycobacteria, what their cell wall looks like. All right, any questions about the atypical cell walls? If not, you can damage the cell wall in a bacteria, and if you do damage it, you will make the cell susceptible to osmotic lysis. I've already mentioned that penicillin inhibits the peptide bridges in peptidoglycan, and that can damage the cell wall. You could also remove the cell wall by using the enzyme lysozyme. It actually digests peptidoglycan. And you don't really need to know that we call the cells different if they're gram-positive and gram-negative once you remove a cell wall. I'm not going to talk about that. I have mentioned a little bit that the gram-negative bacteria are not as susceptible to the gram-positives to various things like penicillin G and certain detergents, because they cannot get through the outer membrane of the gram-negative cell. That doesn't mean that the gram-negative cells are not sensitive to antibiotics. They just are sensitive to other antibiotics, like an antibiotic that can penetrate the outer membrane. Streptomycin is a lipid-soluble molecule, so it can go right through the outer membrane of a gram-negative cell. And then there are even members of the penicillin family that can get through the porin molecule in the outer membrane. For example, ampicillin, which is one of the early members of the family of penicillin, can get through the gram-negative cell wall and inhibit both gram-negative cells and gram-positive cells. Any question about any of that? All right, you guys are quiet, so I'm moving on. Let's talk a little bit about the plasma membrane. I mentioned that we'd talk about that later. The plasma membrane is mostly made up of phospholipids, and they arrange themselves in this orientation shown here, where the phosphate heads face the inside of the cell, because that's where mostly water is. The cytoplasm is mostly water, and the heads want to be facing the water. The fatty acid tails are on the inside of the lipid bilayer because they do not want to face the water. They want to be around other lipid-soluble molecules, and so they're on the inside of the lipid bilayer. And then on the outer layer, the phosphate head is facing outside the cell because outside the cell is mostly water. And this is a bacteria, but if we're going to talk about we can talk about bacteria in the blood. The blood is mostly water, and so the phosphate of the bacteria cell membrane will be facing the outside of the cell because it's mostly water. Any question about any of that? Phospholipid molecules are the main molecule in the cell membrane, the plasma membrane, but they are not the only molecules in the plasma membrane. You can't have carbohydrates and proteins. The proteins are important, and let's talk about three of them. There are peripheral proteins, and they are a protein which are on one side or the other of the membrane. That's why they're called peripheral. There are also integral proteins and transmembrane proteins, and both of these two proteins span the membrane. This protein looks like it is a transport protein, allowing a molecule to either enter the cell or leave the cell. And we talked about that. For every molecule we want to enter the cell, we have to have a transport protein to bring that molecule into the cell. For example, glucose and fructose can only enter the cell through a protein. Any question about any of that? All right. The plasma membrane follows the fluid mosaic model. The fluid mosaic model states that the membrane is a mosaic of phospholipids and proteins. And it states that the membrane is as fluid as olive oil. If you look at my hand here, I'm an animal, so all I have are cells, and they're connected by cell membranes. So what you're looking at here is cell membranes. It always amazes me to think that my hand is held together by a membrane which is as fluid as olive oil. Any questions about any of that? The proteins and the phospholipids can move around in the fluid membrane. The phospholipids can obviously spin this way, but this one can move over here and then move over there. And the proteins can also move. Those of you who have had anatomy and physiology, you know that when a ligand binds to a receptor protein, that those proteins tend to move together before they're brought into the cell. Okay? So the proteins can move around in the membrane. There are only a few exceptions. Is that true? The exceptions are when the protein is anchored inside or outside the cell. If the protein is anchored, like this protein, let's say this one here, is anchored to the cytoskeleton, we'll talk a little bit about that later. It's a membrane of proteins running throughout the cytoplasm of the cell. If that protein is anchored to the cytoskeleton, it's not going to move, okay, because it's anchored in place. The protein can also be anchored in place to the matrix outside of the cell. Let's talk about a bone cell. It's inside bone, and the protein could be anchored to the whatever bone is made up of, minerals of the bone. Okay, any question about any of that? The membrane shows selective permeability. What this means is that it allows some molecules to pass, but not other molecules. Large molecules cannot pass the cell membrane without the help of a transport protein. Ions either do not pass the cell membrane, or if they do pass, they pass very, very slowly. Lipid molecules can pass through the cell membrane because they can dissolve in the fatty acid tails of the lipid bilayer. But if you think about it, there aren't too many lipids floating around in your blood. The blood is mostly water, and it contains mostly water-soluble molecules. So my point is, cells aren't going to see that many lipids. Small molecules can pass the cell membrane. I'm not going to talk about water, but I will talk about oxygen. Oxygen and carbon dioxide can move through the cell membrane by diffusion. Oxygen will move into the cell, moving from areas of high concentration to areas of low concentration. And carbon dioxide will move out of the cells, moving from an area of high concentration to an area of low concentration. Any question about any of that? In the cell membrane, we can also find some special things, like in bacteria, we have the enzymes for making most of the cell's ATP. And we'll talk a little bit more about that when we're talking about aerobic respiration. But where the majority of the ATP is made in a bacteria is on the cell membrane. Enzymes attached to the cell membrane. In the photosynthetic bacteria, we will find the photosynthetic pigments in the cell membrane. And we call these either chromatophores or thylakoids. Of course, in eukaryotic cells, we do not have these in the cell membrane. ATP production happens in the mitochondria, and in photosynthetic plant cells, this happens in the, drawing a blank here, the chloroplast. Any question about any of that? All right, the cell needs to have an intact plasma membrane, otherwise the cell will leak contents of the cytoplasm out of the cell, and that would kill the cell. And that is actually what alcohol, the quaternary ammonium compounds, and the polymyxin antibiotics do. They put a hole in the plasma membrane, allowing the cytoplasm to leak out of the cell, and that will kill the cell. Any questions about any of that? All right, materials move across membranes according to the law of diffusion, and that is, materials will move from areas of high concentration to areas of low concentration. The movement of material across the plasma membrane is critical to cells. Cells can move material across the plasma membrane in two different ways. There's passive, and that's where the movement occurs down a concentration gradient, meaning the material moves from an area of high concentration to an area of low concentration. This is passive. We'll talk exactly what we mean in a little bit. That can happen by simple diffusion, where a solid will move into the cell. And the best example of that would be oxygen being dissolved in the water can move into the cell by simple diffusion, a passive process. Passive processes cost the cell no energy. Molecules can also move into the cell by facilitated diffusion. Facilitated diffusion uses the laws of simple diffusion, but it moves across the membrane through a protein. But once again, the movement happens down the concentration gradient of the molecule, and the cell spends no energy. You can also have osmosis moving into the cell. Osmosis is water, and when it moves across the plasma membrane, we call it osmosis. Osmosis is different because the movement of water is critical to cells, and then water is not as solute. When we're talking about simple diffusion and facilitated diffusion, it's the movement of a solute. Osmosis is the movement of water, and water is a solvent. But if water was a solute, it would move by, well, largely by simple diffusion. Okay? We'll talk a little bit more about that later. The other way that cells can move material across the cell membrane is an active process. The molecule is moved against its concentration gradient. And let's say we have lower glucose outside the cell, but the cell wants the glucose, and so it will move it into the cell by an active process. This will require a transport protein, and to move one molecule into the cell against its concentration gradient, that will require the energy of 1 ATP. Any question about any of that? All right, let's quickly go through diffusion. Simple diffusion is just the molecules of a solute from areas of high concentration to areas of low concentration. And that is how oxygen and carbon dioxide move in and out of a cell by simple diffusion. Facilitated diffusion is simple diffusion through a membrane, and it requires a protein to move the molecule across the cell. But the molecule is still moving by the laws of diffusion, meaning in the case of fructose, it moves into the cell by facilitated diffusion, and fructose will move into the cell from areas of high concentration to areas of low concentration. Once the fructose concentration inside the cell equals the fructose concentration outside the cell, you will get no net movement of fructose into the cell. And that's important because if you're eating fruit and getting fructose and you're trying to watch your weight, it's good to know that fructose, not all of it will be brought into the cells and then digested. Okay? Any question about any of that? With glucose, when glucose is higher outside the cell than inside the cell, glucose will move into the cell by facilitated diffusion. However, once the glucose levels equal outside the cell and inside the cell, this protein will change, and it will no longer act as facilitated diffusion. It'll act as an active transport protein, and then that protein will pump glucose into the cell against its concentration gradient. And for each, did I say fructose? I meant glucose. For each glucose moved into the cell, it will cost the cell 1 ATP of energy. But the cell wants the glucose, so it will move every glucose out of the cell into the cell. And that's good to know if you're watching your weight. Anything you eat that's sweet, everything will be digested. All the glucose will be digested and absorbed. Okay? Any question about that? And so, yeah, this picture, which I wasn't showing you, the glucose port, it's not exactly correct, and that is glucose only moves into the cell by facilitated diffusion when glucose is higher outside the cell than inside the cell. And then this protein changes and it'll act as an active transport protein. Fructose doesn't have the change, which is why I talked about it. All right, so the passive processes move things down its concentration gradient, and that happens in both simple diffusion and facilitated diffusion, where solutes are transported. Osmosis is the net movement of water across the selectively permeable membrane, and water will move from areas of high concentration to areas of low concentration. This we do not call diffusion, we call it osmosis, because it's the net movement of water. You should realize that the tonicity is not defined on the concentration of water. The tonicity is defined by the concentration of the solute, but in the case of osmosis, it's the water that moves. Oh, and I can also say that, oops, wrong one. Wherever you have, oh, I went way too far. Wherever you have a high water concentration, you have a low solute concentration. Wherever you have a low water concentration, you have a high solute concentration. All right, so a cell can find itself in 3 osmotic solutions: an isotonic solution, a hypotonic solution, and a hypertonic solution. Note that these are relative terms, so you should say that the solution outside the cell was hypertonic relative to the solution inside the cell. In an isotonic solution, the concentration of solute outside the cell is equal to the concentration of solute inside the cell. There will be no net movement of water into or out of the cell. And the cell will be happy because it's in an isotonic solution. It doesn't have to worry about bursting if it's lacking a cell wall. And our red blood cells, when they're in the blood, they are in an isotonic solution. Any question about any of that? The cell can also be in a hypotonic solution. That's where the solute concentration outside the cell is lower outside the cell than inside the cell. That means water will move into the cell. And if this cell doesn't have a cell wall, water will move into the cell until the plasma membrane bursts, and that will kill the cell. Most bacteria live in a hypotonic solution, and that's actually why antibiotics work. All right, the last one is a hypertonic solution. The concentration of solute outside the cell is higher than solutes inside the cell. That will result in the movement of water in the cell moving outside the cell. This will cause the plasma membrane to pull away from the cell wall, and it will lead to plasmolysis. For many bacteria cells, this can lead to death, like Streptococcus pyogenes, which causes strep throat. If this were to happen to the cell, it would die. E. coli, it will lead to death, although I'm not sure how long the E. coli can survive this way, but it will kill the E. coli cell. For cells which love a highly saline environment, they can survive this process, like Staphylococcus aureus can survive a high salt concentration, and it can live in 20% sodium chloride on your skin. All right, any question about any of this? If not, oh, I've got a 2 here already there. That's where I'm going to stop, and we will continue with this lesson at 6:30. Are there any questions? If not, I'll see you at 6:30.
Thank you.
All right.